Very interesting. When I don't give the introduction and you do your own research, and I hear them like, hey, I forgot that I have all of these distinctions. Uh, So it's always an absolute honor and a pleasure to be here. Thank you to the team for inviting me. It's been ages. I've been in touch with the team for years now, but it's finally come to fruition that I could be here on the days and talk to you all. Um, the thing is, all said and done, what I would like to be taking you through is a small journey of not only me, my work, but also a small journey through something that I believe is pertinent to life on Earth, and that is what we'll soon figure out. So let me start off with this quote by Sally Ride. Sally Ride was a teacher. She was an elementary school teacher in the United States, and she was chosen as the person who was supposed to go on the Challenger expedition. Unfortunately, that's the expedition that also blew up on taking off, so we lost a great person, but this is a quote from her. Science is fun. Science is curiosity. We all have natural curiosity. Science is process of investigating. It's posing questions and coming up with a method. It's delving in. So that's what we do. That's what I do. We ask questions. We are curious. The reason I am a scientist, the reason I am standing in front of you on this day is because I was a very, very annoying kid. Because as a kid, I used to keep asking why, what, how, when. Whenever I met my mom and dad, what is this? Why is this? And they always ended up introducing me to the encyclopedia or shifting me off to my grandmother and grandfather. And that incorrigible curiosity turned into my love for science and my love for finding out answers. And that's how I ended up where I am right now, which is a senior scientist. Now, the thing is, as you would have heard, there were two organizations that are used to my name. On the left is the Dystrophy Annihilation Research Trust, which really conveniently acronyms to DART, D-A-R-T. On the right is another organization called Hanugen Therapeutics. I will come to it when I come to it and talk to you about why there are two organizations are there and what we do. But before that, let's take a small trip. So in 1953, the following sentence appeared near the end of a paper, Nature 141, Sound 37, Sound 38. Okay, obviously all of you do references. You see that very interesting thing? It says 737 to 738. What does that mean? It means the entire article is just one paper, one A4 sheet. Single A4 sheet is the article. Remember how we write 30 pages and give it to our HOD and HOD said, the paper or not. Yeah? This is an article which is literally just one page long. At the end of the page, there's this one sentence at the end which says, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Now, according to me, this is it's the biggest understatement ever in the history of biology. So obviously you know who these two people are. That is Watson and Crick. And on the lady on the right is Rosalind Franklin. And that paper was the one that told us about the double helical structure of DNA. And by that one sentence they put up that specific pairing we have postulated suggests possible mechanism that single-handedly told us everything that we need to know about this molecule called DNA. Now I make a point to talk about Rosalind Franklin because in 1953 she was not given any due respect. She was not given any tribute. Her name was not on the paper when these two won the Nobel Prize. They did not mention her. She did not get any money out of it. But she's the one who's responsible for this. Many years later, now there's a movie coming out starring Nicole Kidman about Rosalind Franklin. I would highly suggest you go check it out. It's fantastic. So I hate to say this, but that was 1953. It's 2022 now, close to 70 years, and still the role of women in STEM, thank you, Avinash, has not, well, it's not grown by leaps and bounds, but still it's much better than how it was. So any opportunity we take, given, we should give credit where credit is due. especially to women in STEM. So this is my small shout out to Rosalind Franklin. Thank you, ma'am, for the work done. And because of you, we are here what we do. To Rosalind Franklin. So the double helix. It's really beautiful. It's a wonderful thing. First time in nature that we come across a double helix. But what does it mean? It means everything for inheritance. Okay, let's take a small detour. So this is Richard Dawkins, a wonderful evolutionary biologist. So in 1976, he wrote a book called Selfish Gene. And he coined a term for discussing evolutionary principles, especially as a mode of cultural phenomena shared from one place to another. It refers to genes, that they self-replicate, mutate, respond to selective pressures. Okay, they have to self-replicate, meaning they can grow. They can mutate, meaning one can become another, and respond to selective pressure, meaning if there is positive selective pressure, they will grow. If there's negative selective pressure, they will not grow. Now, here's a question to all of you. What is that word which Richard Dawkins came up with, which means something that can self-replicate, mutate, and respond to selective pressure? I'll give you a clue. All of you know this word. All of you have seen it in social media, but most of you, or probably none of you, are aware that it comes from the word of evolutionary biology. But it's the same thing. It's something that replicates. You can make changes to it. Somebody can make a change to it. If you like it, you send it forward. If you don't like it, you delete it. But if it's something that's really good, that's really positive, then you send it to everybody. Now, can somebody tell me what this is called? Something on WhatsApp. Something on WhatsApp. I'll give you another clue. It's just 4 letters long. Something that all of you know. If you like it, you forward it. If you don't like it, you don't forward it. So it doesn't grow. Post, feed, then text, chat. Okay. Something else which encompasses all of this. It's just 4 letters. You would have come across it on Facebook, Instagram. Yes. Who said that? Somebody said, well done. What's your name? Can we have a round of applause for Sangeeta? This is what we call a meme. M-E-M-E. Do you guys know what a meme is? I'll give you for example of a meme. So this is a meme. So this is called the show me power meme. So show me power Duracell, show me real power, 3 stacks and radiative power technology. Then he says finally perfection, and you have a picture of the mitochondria because mitochondria is the very good, excellent, 10 points. How about this meme? I wish I was 39 so I could get paired with you. It's a genetic pickup line for boys and girls from genetics who go to a party and want to figure out how to date a boy and girl. Okay, how about this? This is a really nice meme. This is about plants making oxygen. So as you can see the lady over there, she's got lots of microphones. She stands for trees. But then there's this poor fellow who's oceanic plankton who's actually done all the work, but they have only one microphone for him. So this is the meme which explains that in the world, oxygen is made up of 80% of oxygen is produced by oceanic plankton. But nobody gives a shit. Like, okay, oceanic plankton is something in the ocean. Everybody talks about trees. Amazon rainforest. Wow. But the oceanic plankton. So, but nobody gives it any, don't look at it. Here's another one from Game of Thrones. Can you show DNA and RNA visually? And this is a visual representation of RNA and DNA. Those who know, those who don't know, I'm sorry, sounds very weird. So these are memes. And the concept of memes comes to us from Richard Dawkins from the world of evolutionary biology. And it's a great way of, sharing information. And like you see this, you like it, you will forward it, right? If you don't like it, you will delete it. The same way is your genetic analog for evolution. So if there's a specific character or a genetic analog which grows because of positive information feedback, then will keep growing. If there's negative information feedback, it won't grow. And you can use the same template and you can make your changes to it. So that is mutation, right? So these are the different things that would happen, a meme or a gene or DNA, when it comes to inheritance of knowledge. basically data going from one place to another, which brings me to my organization. So the Dystrophy Analysis Research Trust, or DART, was started to achieve total annihilation of muscular dystrophy, and it was started by these two people, Anand and Mohan, and that's the kid. His name is Karan. He's wheelchair bound because he suffers from or suffered from Duchenne muscular dystrophy. My apologies, I'm just coming down from a cold, so any interruptions, my apologies. So, when DART was started, it was started by parents to help parents to tell them about what is DMD, what is a rare disease, what happens when you have muscular dystrophy, so on and so forth. Basically, our awareness initiative. Then I joined DART, and I joined DART as a science person. So I was the first non-NGO, non-awareness person, and we started a scientific organization. So let's talk about DMD. Now, unfortunately, there's a lack of awareness about Duchenne muscular dystrophy. So let's just get that out of the way. It is a muscular disorder which is evident from the age of 2 or 3. As you can see from both the pictures, you can immediately visually identify a kid who's suffering from muscular dystrophy. The way that they stand, the way that they walk, and the way the spine is slowly deformed with age. So basically what Duchenne muscular dystrophy does is it starts compressing the body from inside on both sides, so that like a accordion you get compressed, and the kid starts walking on his toes, and sometimes what happens is yes, thank you so much. So as you can see, there are visual indicators of muscular dystrophy. Now, this is an X-linked disorder, meaning it's found only in the X chromosome. On X chromosome, on 21.3, you'll see you have the gene for dystrophin. I will come to that. But what is X-linked inheritance is basically because it's found only in the X chromosome, it is also carried over by the mother. Now, what happens if a son gets the affected X chromosome from the mother? That means he starts showing symptoms for DMD. If he gets the X from the father, then it's not a problem because he's unaffected. Now, girls have 2 X chromosomes. So even if one of them gets the affected X chromosome, they have a backup, another X chromosome, which makes up for the lack of the first one. So mostly 99% of the time, women with DMD are completely asymptomatic. So you do not know whether they have DMD or not, but they are carriers. So that is how the inheritance is. Now, obviously, your question is going to be, why isn't there another generation? The reason being, DMD is fatal. So the boys usually do not grow up to be in a position to procreate. The disease affects at a very young age. By the age of 8 or 9, they are wheelchair bound. By the age of 14, they are usually bedridden. And 2021 is when your most important muscles, your heart muscle and your lung muscle, the pulmonary and cardiac muscles, they give away. So it's a fatal disease. So you do not get another generation. Now, how does this happen? Everybody knows the central dogma of life: replication, transcription, translation, DNA, RNA, mRNA, and so on and so forth. One of the things that plays an important role in it is something called the messenger RNA or mRNA. Now, to come to the mRNA, the gene has inbuilt mechanisms to ensure that the right amount of protein is formed, or the right functional version of the protein is formed. Now, DNA has 2 things. It's called exons and introns. So exons are the part of the DNA which hold the valid information that's important for the protein to be formed. The intron are the small spacers between the exon, which disallow the DNA to be read fully, so that it does not by mistake get read. So eventually, when an mRNA is formed, the introns are snipped away, the exons are put together. It's basically like a jigsaw puzzle. And once all the exons are in order, then it is read by the apparatus, and you have a protein at the end. Now, this is what happens to all of us on a normal day. But let's talk about rare diseases. So, what are rare diseases? Now, obviously, rare diseases are called rare because they do not affect as many people as other diseases do. For example, what I am suffering from, what I am unfortunately was suffering from, laryngitis, is not a rare disease. Everybody knows somebody who's got a cold. Everybody knows somebody who's got a fever. TB nowadays also is not a rare disease. Everybody knows somebody at least who have TB. But how many of you know a disease called Duchenne muscular dystrophy? Right? It's quite rare. You've not heard of it. Probably not, at least in a non-academic sense, because it affects only 1 in 3,500 boys. Now, again, 1 in 3,500 doesn't seem like a big number. I'm guessing this college has at least 3,500 students in one building. Okay, and one of them having a disease to us doesn't seem rare. But on a clinical side, it is a rare disease. Now, till 2019, the Ministry of Health and Welfare, India, did not have anything to do with rare diseases. They followed what are known as the guidelines of the World Health Organization, WHO. Now, the WHO makes its statistics according to the status in the United States and in Europe, right? In Europe, 3,500 is pretty much the population of an entire town. But 3,500 is the number of people who live in 3 blocks in Bangalore, right? Like, there are housing complexes in Bangalore, one address where 4,000 people will live, right? So there's not a big number. I mean, rare disease cannot be counted like that. So a whole bunch of us, we went to the government and we fought with them saying you cannot use the WHO guidelines. In that case, rare diseases are just too much, and that also means the funding that has to come to rare diseases is less. So finally, after much fighting, they started the national policy for treatment of rare diseases, and we were sorry, we are very fortunate and blessed to have the government of India wake up to the fact that rare diseases are a huge problem in India, especially genetic diseases, and they brought this out. So let's get back to the dystrophin gene. Remember I told you about the exons? So the dystrophin gene has 79 exons, which makes it the largest in the human body. It's almost 2,400 kb, and the mature promoter site is 14 kb, multiple promoters, and it encodes dystrophin, which forms a structural role. Now, if you see that purple color muscle, that is the role of dystrophin. Basically, think of it as a shock absorber. If you're on a scooter or a bike, you have a shock absorber which is between the seat and the wheel, so that when you go up and down, it does not affect your traveling. So it acts like that. Now, when all 79 exons are read and they are properly translated, you get dystrophin. Okay, but what happens sometimes? In the middle of the 79 exons, there's a small mutation. Small change. A G can become a C, a U can become a T can become an A, somewhere somewhere goes wrong, and there is a small cut. As you can see in this, what we're looking at is between 49 and 51. 50 is missing. So you have 50 here, this double concave looking jigsaw puzzle piece, that unfortunately is deleted. Let's say deletion mutation. So what happens is the mechanism is not able to read the entire line. That unfortunately means the mRNA is also truncated, which means there's not complete translation, and the dystrophin protein is only half its size. Now, what happens when you have shock absorber which is only half its size? It's neither able to absorb the shock or do its work properly, and that's what happens in this case. Now, there are 2 things that can happen. One is the kid has DMD, that's Duchenne muscular dystrophy. The second is Becker's muscular dystrophy. The only difference being that DMD is far more fatal. Death happens at the age of 19 or 20. Because they're ambulant until 16 and live up to the age of 40 or 42. Right? So, what and how does that work? Before that, this is Professor Steve Wilton from the University of South Wales, Murdoch University, Australia, Southwestern Australia. He's an academician and he figured out something called exon skipping. So, what is exon skipping? Remember, I told you exons? So, all exons are basically collections of codons. So, what is a codon? Codon is a three amino, just like ATG or CTG, just three letters bunched together to form a codon, and that is read as a frame. And when you read that as a frame, the DNA, then finally translation, mRNA, is the protein. So, for an example, let's take this sentence. The mad cat ate the fat rat and the big bat. Now, every single word is three letters long, meaning they're all codons. It makes sense. You get the idea? There's some story about a mad cat, it's a fat rat, the big bat, something, each everything. Okay, it's a coherent sentence. Now, let's take that sentence and have something called an in-frame error. Now, an in-frame error, so four of the words get deleted. Fat, rat, and the four words get deleted by some typo. So now the sentence is, the mad cat ate the big bat. Now it is shorter, much shorter, but still makes sense. You can get some information out of it. So this is a truncated sentence. Now let's go for out of frame error. Take the same first sentence, but instead of four words, only F and A get deleted. Just two letters. F and A get deleted. Now, what did I tell you about the reading mechanism? It reads in frames of three. Always reads in frames of three. So now the new sentence is, the mad cat ate the tart and with a big tea, which makes no sense at all. Right? You're not getting any information at all. It is longer, but it's just utter nonsense. So this is out of frame error. And in frame error is you get a truncated version, still makes some sense. Out of frame or not so truncated, but is complete nonsense. So what do we do, or what does Steve Wilton do? He took that out of frame sentence and then just took off whatever is tra, tan, with, and e. Right? It took off three words and an extra letter, which brought it back to the mad cat ate the big bat. Now, here's the thing. You can't add new letters to the sentence. What you can do is, you can delete letters. So he's deleted the letters to make it a shorter, but still slightly functional version. Meaning you don't get the full information, but you get parts of it. So at least you know now there's a big bat that's eaten by a cat. Now, this is the difference between DMD and BMD. So, an out of frame error is responsible for Duchenne muscular dystrophy, where there might be a small mutation, but the effect is that the whole thing is out of chaos. But in a Becker's muscular dystrophy, there might be a larger mutation, but there is more functionality. So, this is Alice in Wonderland, the big mad fat cat. Now, what Steve Wilton did was, he invented the concept of exon skipping, and that in 2016 won a regulatory award from the FDA, who are the US body responsible for basically any new drug on the scene. And it became a revolutionary thing. Now, the thing was, he was an academician, and the entire of Australia, they had about 13 patients. We, on the other hand, in Karnataka alone, I'm not even talking whole India, Karnataka alone have about 2,000 patients. Okay, that goes to show you what is the extent of these diseases in India. The reason we do not know is because of lack of awareness. We had more cases in the city than they had in their entire continent, which meant that they did not have enough samples to do studies with. So that's why they worked with us and we worked together. So these are the 79 exons. Now, to help you visualize how it works, there are three different types of bonds. You have the flat bond, the concave bond, and the convex bond. Just to help you understand. In real life, they are more like actual bonds. So now, as you can see, at some places, they're all the same. Like if there was a mutation here, it wouldn't matter at all. Let's say 33 gets deleted, 32 and 34 would still go ahead and join up. So you'll have an in-frame error. So not a problem at all. But let's say 45 gets deleted. Now 44 is concave, whereas 46 is flat. So the reading mechanism will keep reading, but stop here. So what Steve decided is, okay, if a 45 is missing and 44 is not going into 46, why don't we take out 46 ourselves? So we physically take out 46, which means 44, which is concave, and 47, which is convex, can go into each other. Which means, although the entire thing is now shorter by 2, it is still fully functional. The reading mechanism does not stop here. Because 41, 42, 43, 44, 47, 48, 49, 50, it won't stop. Now, what does this mean for us? What this means for us is that we are not curing the disease. The kid is born at DMD. The levels of dystrophin in his blood are steadily dropping as he grows older. We can't do anything about it. What we can do is we can stop that decrease at a particular age. The earlier that we do an exon skipping approach, from that moment onwards, the amount of destruction of dystrophin or the amount of reduction of dystrophin comes to a halt. Again, this is not curing. We cannot cure the disease. We cannot put back dystrophin. We can't make the gene start making dystrophin. But we can stop the gene from not making zero dystrophin. Okay? We all have 100% dystrophin. These kids have 20%, 30%. What we are doing is they go from 80, 70, 60, 50, 40, 30. If we stop them at the 60, then it's going to be 60% throughout. It doesn't go to 50, 40, 30. What that means is you're doing two things: adding to the quality of life and adding to a length of life. What a kid would have probably died at the age of 21 or 22 is now going to live to 28 or 29. So we do two things. First of all, we take a skin biopsy, right? We take a fibroblast. We don't do muscle biopsies because the kids already are lacking in muscle. It doesn't make sense taking muscle from them. So we take a fibroblast and we use adenovirus and we force it to become muscle cells. This is called forced myogenesis. And then using transfection, we have two ways of introducing the compound into the cell. Now, thankfully, zebrafish, Dania rario, are one of the very few animals that can show DMD. Unlike other animals, where it takes years and years for it to show, in 2 weeks a zebrafish can show, and it doesn't do anything to their lives, just as a small crick in the tail. So ethically, it makes sense to use a zebrafish. So we do that, and then we do obviously efficacy studies on artificial, Western blot. Then we do toxicology studies, which is, as you would know, increased dosage, toxicity, slowly we increase the dosage. Please keep bringing it up. And then we have something called an MDX mouse. Now, an MDX mouse is a mouse that is bred genetically with a 23 exon mutation. It is born without the exon number 23. So we can test our compound on 23 or 24 exon skipping method and test it on the mouse. Now, what is the problem with this, and why was this not done, or why do we have to wait for this long? Because every single kid has his own mutation, right? Because all of them have different DNA and everybody has a different type of mutation. So each compound that we do is custom made to that patient. This is personalized genetic medicine, right? We cannot make one drug and give it to 3 or 4 people. Even siblings, we cannot make the same drug for siblings, even if they suffer from same type of DMD. It is not possible because we have to make a custom-made, personalized genetic medicine. Now, this was one of the reasons why, as latest as 2018, there were no clinical trials for custom genetic exon skipping in India, right? The biggest, I think, was the United States with 19, and then you had the United Kingdom with 14, but India had 0 because of certain red tape and rules that the government had to put us through. Because according to at that time, the Indian Council for Medical Research, the DCGI, they had very specific guidelines for clinical trials. Clinical trials phase 1, you should give the medicine to at least 200 different patients. You should also give a placebo. You should also give the same thing to somebody who's not suffering from DMD and make sure there are no effects. Now, you see where the problem is? This is personalized medicine. I cannot find 200 people with the same gene. I cannot find 200 people with the same mutation. Also, if I were to give my compound to someone who's normal, let's say Avinash, I'm hoping you're normal. Yeah. So let's say we give him the exon skipping medicine, the compound will go and snip off that particular exon in his body, thereby inducing Duchenne muscular dystrophy in him, which is unethical, illegal, and I don't think Avinash would want to do that, right? So that was a problem, and we put this in writing, and we spoke to the government of India, and finally, in 2019, they said, Seripa, let's get back to the point, and they allowed us to do a single investigator-led study. So this was the first time in the history of India that we could do a personalized genetic medicine gene therapy clinical trial. We can't call it a clinical trial because the number of people were there, but it was an investigator-led study. One kid, with his permission, with his parents' permission, and with the entire might of the Indian judiciary behind him, watched us as we gave one injection on IV saline to that same kid who was there on my first slide. So that's Karan. In November 2019, he became India's very first exon skipping genetic trial. He looks very happy because we told him we're going to give him nice Bukhari biryani after this. So he was looking forward to that. And this was the very first time. It was a slow saline. I think for 2 hours, we were really, really careful. And we had to make sure we followed all the markers, cardiac markers, pulmonary markers, weight markers, everything. It was one of the most strenuous parts of our lives, but for 3 years we've been fighting and we finally did it. And this was in 2019, and the 3-year study, which obviously came to a close in 2022. And thank you. It is quite something historical. I'm very proud of what we did. And we did it. So yeah, we were able to prove comprehensively that not only our compound worked, because our cardiac markers were stable, pulmonary markers were stable. I mean, as a scientist, I am looking at all the data, all his pulmonary markers, all his cardiac markers, his dystrophin production levels. That's what I'm looking. But as a human being, I'm also looking at Karan, who now, when he was born, doctors told him, you won't go to school, you'll probably not finish school, you'll never be able to, pass the age of 16. Well, This was in 2022, last year. He became a BSc graduate from Srinivasan College. He finished his undergraduation in biochemistry, and he and I spent 2 hours, 3 hours every day playing Red Call of Duty and NFS on the video game. Now, you might ask me, Bertie, what is a big thing if he can play a video game? He also plays a guitar. I was his guitar teacher, so I taught him guitar. Now, for a person from outside, me, a scientist, playing 2 hours of video game with one of the patients doesn't make sense. Or me sitting with him and playing guitar doesn't make sense. But what you don't understand is one of the biggest problems with DMD is that all your hand muscles and your wrist muscles are the first to go. Now, we had to figure out a way to give him physiotherapy without him understanding or him figuring out it was physiotherapy. And we discovered that all our kids love playing video games. So we gave them all Xboxes, and every single Saturday we'll get together. It was basically 2 hours of physiotherapy, but 2 hours of playing video games and then playing guitar, right? And this was a form of physiotherapy. And we could also track it. This is very important. Earlier, Karan could play up to 45 minutes. After that, he'll be like, I'm tired. And I could see that his reaction time was slow, that his reaction time was fading. After about a year and a half, he was playing 90 minutes, 100 minutes. His reaction time was the same, right? And he was faster with the joystick. So we could use the Xbox as a scientific tool to measure his thumb movement and how it is, which is just exciting. Also, I was able to convince my boss that he has to pay for me for 2 hours of playing video games on Saturdays, so which is pretty cool. And that's what happened. Now, this is where, as you can see so far, on the PPT on the left, on the top, you had a logo which says DART. Now, DART was an NGO. I told you, it started by parents of children who have DMD, which also means we can't do certain things. First thing being that we can't make a profit. We can't make money because we are a not-for-profit, non-government agency. And we had an amazing technology. We had the brains, okay, not me, but everybody else in the lab. And we have all these, and we wanted to show it to the world. That's when the Karnataka government said, okay, we'll help you make a startup. And that startup, now the new logo on the right, came from 2018, which is Hanugen Therapeutics. So Hanugen Therapeutics is a startup, which is started by us, where we gave all our services, all our equipment. We have a GMP facility which has the ability to make personalized antisense oligonucleotides from scratch, all the way downstream to lyophilized powder, which we were using only for our kids. Now we decided to open it for everybody else, and that became Hanugen Therapeutics. And in 2018, we won a couple of. We went on to win multiple awards. We have the big startup, the BIP startup, KSAP, seed startup, and we won pretty much every award there is for grants. And right now, we are one of India's top, I'm supposed to say one of, but I know for sure that we are the only one who are able to produce GMP grade, clinical trial grade antisense oligonucleotides for human trials. So that is Hanugen Therapeutics. Now, this is a very, very new development. As you can see, this is from 29 December 2022. That's just last month, less than a month back, or exactly a month back. Very interesting. Yeah. So exactly a month back, the government of India, backed by the High Court, put out a actual on record that the government of India has to pay 5 crores to Hanugen to set up a clinical trial across the country, which means now the government is invested in getting rare diseases the due justice. So now we have been asked by the government to produce clinical trial based antisense oligonucleotides for a clinical trial for across the country. Now, this brings me to the place where I should say, yeah, a rare disease, it may be rare, but hope should not be. I am 100% sure if you go out today, you do not know this, but you are meeting somebody who has somebody in the family who suffers from a genetic disease. They might not know it, or they might not be comfortable saying it out. But the number of people who suffer from rare disease is much higher than the whole number of people who suffer from infectious diseases. The reason being, obviously, we have a very high population, one, and we have very tight genetic pools, which means our gene structure itself has become progressively more tighter, so that genetic diseases which used to be recessive are now dominant. Simple thing being diabetes. If you know about diabetes, you know there's a diabetes called MODY, maturity on diabetes in young. Okay. At one point of time, it's a mutation of the HNF4 alpha gene. It used to be a really rare disease. Not anymore. You get about 10, 15 patients every other week at the diabetes situations. So rare diseases are not rare, and they're becoming increasingly not rare. And the problem is you have so many conditions, millions of people suffering from them, and there is a distinct lack of any sort of treatment. Why? Because companies can't make money out of it. Let's say Procter and Gamble want to get into the DMD scene, and they'll say there's one kid who has DMD, so they have to spend thousands and thousands of dollars, probably $1 million, coming up with a medicine for one kid. And can they sell it back to that kid? They can't, right? So how will you make money? So any pharma company will not look at rare diseases because they cannot make money. That's why we had to do this. That's why the parents had to get into it. That's why we are writing grants ourselves. And that's why the government is stepping in and saying, we will help you, right? Now, as I look out onto the audience, I realize that there are a lot of people who might have not even thought about this as an option. So I'm just letting you know that this is something that you can think about. You can do your thing, you can do whatever you want to do, but in the back of your mind, just remember that there are hundreds and hundreds of rare diseases outside which need our help. As you can see, India has the highest percentage of rare diseases per population, right, in South Asia. Obviously, again, because of our population. But there are multiple rare diseases, and all of them can be looked at and looked into using just very simple genetic methods. Now, that's not to say that we have not done any work. As you can see, with the number of drugs that have been done, the number of papers that have been published in rare diseases, it is growing up. It is a field that is fast growing. We are looking at least $26 to $27 billion being invested in research in rare diseases by 2025. So that's personalized genetic therapy for rare diseases. Now, at this juncture, I have to pay tribute to an important person who's responsible for us coming this far. That is Professor Soumya Swaminathan. Now, you might all know her as the chief scientist of WHO. She was a director of ICMR, and we kept pestering her. At that time, we wanted 30 lakhs, 50 lakhs, and we used to write grants and we used to write mail to her. And she kept hearing about this family, husband and wife, who started a research organization in Bangalore, who say they can not only take care of the child, but can take care of all rare diseases, all this fake news. It sounds very fake news, obviously. If somebody sends you a WhatsApp forward saying there is a husband and wife, their son has a rare disease, they know how to treat it, and they started one multi-crore research organization, it's very fake news. If you send it to me, I'll delete it. But that's how it was. And she kept hearing about it. Then, to her tribute, I mean, absolutely fantastic lady, she decided to visit us to see if it was true or not. And when she did, she realized that yes, it was not fake news, we were actually doing something. And then she further on went to become a huge crusader for us. She was responsible for spearheading that first clinical trials. She obviously went on to become deputy director of WHO, but she still is one of our biggest motivators. And what she did was to change two things. One is how the ICMR looks at clinical trials, and two, how the government looks at rare diseases. And it's thanks to her and my bosses, Anand and Mohan, who started it, that India right now is at the forefront of personalized genetic therapy. We are one out of only 5 countries in the world who have done a clinical trial in genetic therapy. So, yeah, that is your future. And that's where I'm standing right now. I think that's about it. Yeah, next will be my Frankish slide. Yes. that brings me to the end. So thank you all for being an amazing audience. Thank you for the opportunity, and I am willing to take questions.